Hello, this is Alpana Prajapat, an associate scientist at Sartorius. Today I'll be discussing a poster which is previously presented as Society for Laboratory Automation and Screening / European Conference, an exhibition in Hamburg, Germany in May 2025, which is titled Advancing Drug Discovery, Integrating 3D Spheroid Models and Live Cell Analysis for Compound Screening. Introduction. Phenotypic screening, which measures defined cell changes in cellular phenotype in response to a multitude of compounds, is essential in early-stage drug discovery. The study investigates the use of a 3D single spheroid model combined with the live cell analysis for drug screening. A library of 8 FDA-approved drugs was profiled to assess changes in spheroid size and viability. Our result demonstrated the advantages of live cell analysis, capturing detailed biological information and maintaining cell health in a physiologically relevant environment. Comparative analysis between 2D monolayer and 3D spheroid models revealed important insight into compound responses, highlighting the potential for improved in vitro to the in vivo translation. Overall, the integration of 3D cellular assay with live cell analysis offers a powerful and robust tool for phenotypic screening, enhancing drug discovery efforts and ultimately contributing to the development of more effective treatment. So here workflow depicts phenotypic screening here, the use of live cell analysis to collect images that are quantified to provide insight into compounds mechanism of action. A549 nuclide red cells were seeded into 9C6 well, usually round bottom plates. Plates were centrifuged to form spheroid and was monitored to desired size with bright field and HD phase contrast image acquisition every 6 hours using InQSight live cell analysis system. After 3 days, once spheroid had formed, compounds were added from screening library. In total, 22 96 well plates were treated, 2 replicates per compound plate. Compound effect on spheroid growth, shrinkage, and viability were quantified using the InQSight system and analyzed using the InQSight spheroid analysis software module. Integrated intensity is used to reflect changes in both spheroid size and fluorescence to provide a quantification of viability. So data analysis workflow, in which in figure A, to evaluate the activity of compound library, cell plates were analyzed over a 10-day period following a 3-day spheroid formation phase. This allowed for the collection of comprehensive time course data for all test entities. The InQSight software microplate view facilitated rapid visualization of full 96 well time course. Control responses were displayed in column 1, which is vehicle DMSO 0.1%, and in column 12, CMP and CHS, which is cyclohexamide, 10 micromolar, indicating a suitable and robust assay window. The remaining 80 wells showed various compound responses within the plate. In figure B, for both the bright field area, which indicates spheroid growth or shrinkage, and red integrated intensity, which indicates the cell viability, a clear assay window that increased over time was observed in figure B. The response of CMP plateaued approximately 4 days post-treatment for both readouts, but an endpoint of 7 days was chosen to compare compound results. In figure C, controls on each plate were used to calculate a z-value, a recognized measure of assay quality, where values greater than 0.4 indicates a robust result suitable for single-shot screening assay. This highlighted strong separation between positive and negative control values. Across the 22 test plates, the z-value was equal to or above 0.4 for both readouts. So for the comparative analysis across plates, the average of the vehicle wells on each plate was used to define the no effect response or maximal spheroid size, which is 100%, while the average of CMP wells defined this positive cytotoxic effect or maximal spheroid shrinkage, which is 0%. All responses were then normalized to this range, and duplicate plates were averaged to facilitate easy comparison of all compounds. The library consists of compounds targeting a vast range of mechanism of action. To gain an overview of responses, the normalized value were plotted across all the plates for both spheroid size and viability based on average bright field area and red integrated intensity. When the two readouts are compared directly, the region of interest can be identified. Region A represents compound that elicits a response of less than 50% for both readouts, 5.3%, which would be considered cytotoxic in nature, causing reduced viability and spheroid shrinkage. In region B represents compounds that affects spheroid size but have limited effect on viability. Cyclohexamide has a known cytotoxic compound added as a control. Sits in this region, which is red marker. This region could include both cytostatic compounds and compounds that cannot penetrate the spheroid structure to elicit a response. And in region C represents compounds that display an increase in spheroid size, which is greater than 150%, with limited effect on viability, indicating the possibility of spheroid disruption. Further compound insight. Here images taken 7 days post-spheroid formation for various treatment groups. The vehicle image shows a compact red single spheroid with some ruffles around the edges, which has been cleanly masked using the integrated spheroid software. In contrast, the CMP-treated spheroid is smaller and has a reduction in visible fluorescence, defining the assay window. Clofarabine, which targets deoxycytidine kinase and DNA polymerase, mimics the CMP cytotoxic response as shown in both the image and in the time course graph, which is in purple line. Flumazenil, targeting GABA receptors, displays a more cytostatic profile with a similar reduction in spheroid size as clofarabine, but no effect on viability, showing here in a pink line. Both vinorelbine and vinblastine, which targets microtubule processing, displays a disruptive phenotype, causing the spheroid to lose its compact phenotype. This manifests as an increase in spheroid size, which is maximum 48 hours post-treatment, and a partial drop in fluorescence, which may be linked to the increased area of the mask and how the integrated intensity value is calculated. For further investigate responses, selected compounds were tested over a wide range concentration between 0.01 to 10 micromolar in 384 well plate for maximal data collection. Table lists responses for active compounds with target information. CMP, which is in teal box, displays a rapid clear concentration related effect, yielding an IC50 value of 0.63 micromolar. Topotecan and daunorubicin, both targeting topoisomerase isoforms, displays in similar cytotoxic effects. Notably, gemcitabine, a DNA intercalator, resulting in a potent effect, which is 0.06 micromolar. Other active compounds such as PKC412 and amphotericin B, acting through a range of targets, displayed potent effects. Deflazacort, targeting glucocorticoid receptor, was shown a partially effect viability and was not initially identified as a hit for the parameter. Interestingly, 23 glucocorticoid interacting compounds were highlighted as active for the spheroid size parameters. Paclitaxel, targeting tubulin, was highly potent at all concentration tested. The new Incucyte CX3 supports multicolor single spheroid imaging and quantification, enabling both bright field and fluorescent imaging with the use of confocal max projection images. A549 nuclide orange cells were treated with various compounds in combination with Incucyte cytotox green over 5 days. Microplate view at 5 days shows both the cytotox response in green and cells in the spheroid in red. A, which is in the zoomed in images, highlights the cell death starts peripherally. Quantification of the CMP responses, which is in B, shows an increase in green and decrease orange fluorescence over time. So summary and conclusion. In this study, we demonstrated the effectiveness of using 3D single spheroid model combined with the live cells analysis as a robust assay for compound screening. By assessing the activity profile of a compound library, we were able to evaluate various parameters, including spheroid size and viability. The use of phenotypic screening allowed for the quantification of growth and compound cytotoxicity, providing a comprehensive alternative to traditional target-based screening methods. Overall, the integration of 3D cellular assay with live cell analysis offers a powerful tool for drug discovery, enabling the identification and characterization of key compounds with potential therapeutic benefits. This methodology supports a multifaceted investigation, enhancing research and screening efforts, and ultimately contributing to the development of more effective and targeted treatments. So thank you for listening to this short summary of poster. If you'd like to know more information, please visit our website where we have poster available as well as more detailed application notes and more information web pages available. Thank you.